and we are working in the human physiology department of the Tripura University, and our research interest is immunological response of different immune cells like CTLs, NK cells, macrophages, and neutrophils in experimental leukemia model. We developed a leukemia model in animal, specifically in mice, and inducing with ethyl nitrosourea. And after 5 months, we got blast cells in peripheral blood as well as in bone marrow, and we have confirmed it by slide preparation. And also, we are interested to develop some medicine from plant of Tripura. And one of the work we are nowadays working is pineapple from Tripura. And you know that the bromelain is one compound extracted from pineapple, and it is commercially available from different companies. But our research on this pineapple is that we are extracting both bromelain and peroxidase together, who have much more potent anti-cancer activity. And also other different medicinal plants from Tripura we are working to use as anti-cancer property, and also antibacterial, antioxidant, and anti-diabetic properties we are looking for in different bacteria and also different cancer cell line. And here you can see our preliminary data that we are inducing leukemia, and in peripheral blood you have after treatment with ethyl nitrosourea the blood cells. In bone marrow have the blood cells after 5 months of treatment, and we are treating with ethyl nitrosourea 2 times in 1 week interval at 80 milligram per kg body weight. And the activity of the cells when the leukemia developed, specifically 2 types of cells fight against the cancer cells. These are the NK cells and cytotoxic T lymphocytes. And here you can see in leukemic conditions, the activity of lymphocyte, cytotoxic T lymphocyte is reduced. And after treatment with interleukins, recombinant interleukin in mice, IL-3 and GM-CSF, this activity is going up. And also, not only the cytotoxic T cells, the NK cell cancer killing capacity or cytotoxic activity is also going up after treatment with GCSF and IL-15 in combinations. And so from here, we use the interleukin, recombinant interleukin, because we know that when we treat the leukemia patient with chemotherapy, lot of cells die. Most of them are cancerous cells, but normal cells also die. As we do not have a target-specific drug, so we are thinking that if we can use endogenous interleukins or cytokines, that will be much more beneficial and low side effect for this disease. And here also you can see this killing capacity of both NK cells and CTLs depend on perforin and granzyme. And you see the expression of this perforin and granzyme is going down in leukemic conditions. And after treatment with IL-3 and GM-CSF, CTL perforin is going up and it's almost normal. And for NK cell, for granzyme also you can see in ELU treated animals like where you have the leukemia going down, and after treatment it is going up again. And this shows that perforin granzyme are the key proteins which modulate cytotoxic activity of cytotoxic T lymphocytes. Is one of the key proteins. And here you can see the phagocytic activity of macrophages and neutrophil. Here the macrophage phagocytic activity is also going down in leukemic conditions, and after treatment is going a little bit up, and this is significant. And for neutrophil, directly not several reports are available, but some reports are available that neutrophil also have the tumor killing capacity. And we have tried with lymphoma cells, and here you can see in leukemic conditions, the lymphoma killing capacity is going down. And after treatment with GCSF and IL-15, this capacity is going up again and almost normal. So here we, from these experiments, we can understand the status of functional status of these immune cells is going down in leukemic conditions, and after combination treatment is going up again. And you know that protein tyrosine kinase is one of the main factors after modulation or the mutations of these protein tyrosine kinase can give huge proliferation. It has the signaling capacity for proliferations and differentiations. And here you can see in for NK cells, this PTK activity is going down, and after treatment is going up again. And the opposite effect for cytotoxic T lymphocytes, you can see after ENU treatment in leukemic conditions, the protein tyrosine kinase activity is going up, and after treatment is going down a little bit, and though is not a huge significant. But the thing is here, lot of treatment is going on with the inhibitor of the protein tyrosine kinase. But here we have seen both for cytotoxic T lymphocytes and NK cells, their protein tyrosine kinase activity is different. They are behaving different way. And also we have tried with bromelain and peroxidase together for this treatment. And as we generally drink the juice from the pineapple, we have extracted this bromelain and peroxidase, and from this gel you can understand the bromelain and peroxidase both are present in this juice or extract. We have collected from Tripura pineapple. Two types of pineapples are here, Q and wing, and both of them contain this peroxidase and bromelain. And after treatment in cancer cells, we have seen that bromelain and bromelain plus peroxidase, you can see at 50 microgram per ml, the killing capacity is maximum. And not only that, here also you can see this is K562, and this is another one, U937 leukemia cells. In vitro condition, you can see at 50 microgram per ml, peroxidase plus bromelain is giving the maximum killing capacity, and we have tried different concentrations. So, though only bromelain is available commercially, but if we can extract it from pineapple directly, which contains the peroxidase 2, in both maybe the synergistic effect of bromelain plus peroxidase giving the better result, and maybe in vivo condition in future we will try in vivo conditions if we can reduce the load of tumor in leukemia. And here you can see the in vitro treatment, the K562 and U937 cells both are leukemia cell line. After treatment, the morphology change. After they are going for apoptosis, and also fluorescence microscopy. You can see these are the live cells, and all these reds are dead cells. After treatment, you can see B16F10 in melanoma cell line going killed by this bromelain plus peroxidase, and K562 also treated with this bromelain plus peroxidase, and see most of the cells are died. So they have the killing capacity, and also anti-cancer capacity of the Friedelin is another compound extracted from another one plant from Tripura, and here you can see most of the cells are going for apoptosis. And so we have screened several plants from Tripura to see whether they have anti-diabetic, anti-cancer properties, mainly the anti-diabetic and anti-cancer properties. And these plants includes Atrocarpus caplasa, Musanda roxburghi, Cleomel nerniflora, Echinocarpus frutescens, Puthus cathcati, Delania pentagina, Urena lobata, Canavalia gladiata, Monocoria hestate, and Sarcoclamas pulcherrima, and Puzoldia indica, Justicia generosa, Mena spinosa. And these plants are from Tripura, and we have extracted in different fractions, and we have so we are all still going on our research for anti-cancer and anti-diabetic activity. So in future, we want to know, we want to explore the microenvironment conditions of the bone marrow, because bone marrow is the site from where the normal cancer, normal stem cell is transformed into leukemia stem cells or cancer stem cells. So we need to know the microenvironment, specifically the expression of different genes during initiations of the disease, and also different cytokines in these conditions. So the microenvironment of the bone marrow has changed after induction of leukemia in animal model. Also genes responsible to changes this microenvironment like CXCR4, CXCR12 and other cytokine genes. And effect of different CFU, colony forming unit, to transform normal stem cells to cancer stem cells, and study of molecular changes of these immune cells like NK cells and CTLs in myelogenous leukemia, effect of different medicinal plants to improve the immune status of the NK cells and CTLs. These are our future directions, and these are recent publications from our lab. You can see these are the publication with different plants, and also in immune status of different immune cells in leukemia. Thank you.